Okay, let's get started. Good morning, everybody. Welcome to the first highlight seminar of this year, which is co-hosted by the Ellinger Center for Energy and the Environment and the Department of Civil and Environmental Engineering. My name is Jason Ren, and I'm the acting director of the Ellinger Center and professor in CE department. I want to remind you the seminar is being recorded. Today, we are very delighted to have Professor Derek Lovley to give this first highlight seminar on protein nanowires. Professor Lovley is a distinguished university professor in the Department of Microbiology at the University of Massachusetts. Many of us know that he is a visionary leader in environmental microbiology, and his work has inspired many researchers like myself into this field. He is a pioneer and leader in biogeochemistry, bioremediation, bioenergy, interspecies electron transfer, synthetic biology, and most recently, protein nanowires. He's one of the most highly cited researchers and received numerous awards, including what my favorites are Time Magazine's Top Innovators in Environmental Science and Popular Science Best of What's New in Environmental Technology. Professor Lovley is a fellow of AAAS and the American Academy of Microbiology. You know, just on personal note, whenever I run out of good ideas, I always check back Derek's Google Scholar profile to just get some inspiration. Well, without further ado, Derek, we are so excited to have you here, and the floor is yours.
Great, thank you, Jason. I really appreciate that really kind invitation and also the opportunity to share some of our research. So first, let me go to share screen. Great. So yeah, thanks for the opportunity to discuss some of our recent research on microbially produced protein nanowires. So what we're looking at in this upper left-hand panel is a transmission electron micrograph, which shows the microbe Geobacter displaying some of these nanowires that are electrically conductive. And because of their electric conductivity, they have important biological functions that I'll discuss this morning. That includes, it's shown in this blue box, the ability of Geobacter and other nanowire producing microbes to transfer electrons to other species, such as methanogens, as shown here. Also, these nanowires can allow Geobacter and other organisms to transfer electrons outside the cell onto insoluble minerals, such as iron oxide, shown here. So I'll talk a little bit about how we know that the wires are conductive, also some about the proposed mechanism for electron conduction along the wire, which we think is due to this packed column of aromatic amino acids going through the center of the wire. Then discuss some of the technologies that come from living microorganisms that produce nanowires, and this includes microbial fuel cells, which can produce small amounts of power in some niche applications. A great application is one known as Electrobioremediation. We can also use this property in environmental sensing, and also in some cases for converting carbon dioxide to fuels and other organic commodities. And then finish up with what I'm really excited about now is no longer living with or working with the living microorganism, but rather growing up mass quantities of these wires using the E. coli chassis that we constructed, shearing the wires off the cells, and then employing these wires in as the electronic component in various electronic devices. For example, sustainable energy harvesting, producing electricity from atmospheric humidity, some biocompatible electronic devices, and also biomedical wearable electronic sensors. So I'm going to talk a fair amount about Geobacter because this is where the protein wires were first discovered. So here again is that cell of Geobacter. Here are the wires emanating from the cell. They're about 3 nanometers in diameter and typically 10 or up to 20 microns in length. So they greatly extend the electronic reach of the cell. But I'm talking a lot about Geobacter, but I want to point out that we now know that nanowires are found in a wide diversity of microorganisms, not only bacteria, but even some archaea. And I'll describe how we discovered all these other wires, and just from some general principles we know about nanowire conductivity. So to start though with Geobacter, so this is a microbe I've been studying for some time in my lab. This is a scanning electron micrograph which shows Geobacter colored in green. This brown material is iron oxide that it uses as its electron acceptor. And I like this photo because it shows the physiological challenge to Geobacter. It's oxidizing organic matter to carbon dioxide, so generating or releasing electrons from central metabolism. And somehow it's got to get these electrons from inside the cell, outside to the cell, onto this insoluble iron oxide. And we're interested in the iron oxide reduction because it's such an environmentally important process. So you can see this process taking place in many soils and sediments. So on the left is a aerobic soil. You can see that reddish brown color. That's due to the abundant iron oxides that are present in most soils. When these soils go anaerobic, microbes like Geobacter become important, so they reduce that iron oxide, so that strips the brownish reddish color, leaving this gray or sometimes black color behind. And this is important in the iron cycle, of course. It changes the solubility of iron from insoluble oxide to soluble iron, and that has important implications for the geochemistry, but it also impacts on the global carbon cycle, just because there's so much iron oxide in soils and sediments. And also iron oxides will bind nutrients like phosphate and also trace metals. So when iron oxide gets reduced, it releases the nutrients and trace metals. So there's a lot of biogeochemical impacts on iron reduction. So that's the case in many soils and sediments with which we're familiar, but it also takes place in other extreme environments. For example, very hot environments around hydrothermal vents. One of the most highly conserved capacities of microbes growing at, other than their ability to grow at high temperature, with these hyperthermophiles, is their ability to use iron oxide. This iron oxide reduction is also important in contaminated environments. So this is an example from a jet fuel spill site. On the left, this is an uncontaminated soil. You can see again that brownish reddish color. On the right is a sediment from a jet fuel, within a jet fuel plume. And what's happening there is that Geobacter is capable of oxidizing various petroleum contaminants, including aromatic hydrocarbons, even benzene, to carbon dioxide with the reduction of iron. So that will naturally happen, but once you understand that that's going on, it's possible to devise various strategies to speed up that cleanup. Another type of environmental remediation that iron reduction is useful for is for some toxic or radioactive metals. So uranium would be an example. Microbes like Geobacter growing on iron in the subsurface, if they see the presence of a metal, let's say like uranium-6, which is oxidized and soluble, it will reduce that uranium-6 to uranium-4, which is insoluble and precipitates out. So this is a useful bioremediation strategy. This is a Department of Energy study site in Colorado. There's uranium contamination in the groundwater all throughout here. It's moving towards the Colorado River, but you can prevent its migration just by simply pumping acetate, which is a great food for Geobacter, into the subsurface. So it precipitates out the uranium, as shown here, and prevents its further mobility. So with all that importance of iron oxide reduction, we're again back to we were interested in this basic question of how do electrons generated from central metabolism inside the cell get outside the cell onto these insoluble iron oxides. And one of the, there are multiple mechanisms for this, but the concept of this unique strategy of nanowires really started with an observation that was made by Susan Childers some time ago, in which she noted that when you grow Geobacter, this was Geobacter metalli reducens, on an insoluble electron acceptor like iron oxide or manganese oxide, it will produce, these are flagella to help it swim, but if you look closely, it also produces these type 4 pili. It does not produce any filaments when it's grown on a soluble electron acceptor such as iron citrate. And we think we know what the flagella are for. It helps Geobacter swim around and find iron oxides. But the question of what the potential role, if any, of the pili was not as clear, but we could see all this expression of the pili. So here they are in a blow up here. You can see there's probably literally hundreds emanating from the cell here. Why was Geobacter producing them? And we could see the upregulation of the pil-A, which is the monomer gene for the pili production. And anytime we ever tried to get Geobacter to reduce iron better, we always just saw this was associated with more pili production. For example, this is literally tube after tube of transfers on iron oxide, and it just starts making more, it expresses as many pili as it probably is humanly possible. So knowing that, and then another key observation was made by Gemma Reguera. She noted that when you grow Geobacter on nano iron oxide crystals, the iron oxide tended to associate with the pili. So this was a bit of a quandary. How could the electrons from inside the cell get all the way out to this iron oxide at distance and stuck on the pili? So the hypothesis was, well, maybe these pili are electrically conductive. That actually didn't seem that likely. Proteins are typically insulating or non-conductive. But what Gemma and her collaborators, Kevin McCarthy and Tina Matev, found out was that if you knock out the gene for the pilin production, it eliminates the capacity for iron oxide reduction. And also, Kevin showed with atomic force microscopy that if you touched a conducting tip from an atomic force microscope onto the top of one of these pili, a current would flow through it. That pili or pilin gene deletion, there were some questions about that. So I think I just want to throw in this slide, it came much later, but it just also showed that if instead of just stopping the production of pili altogether, we insert a synthetic pilin monomer sequence that gives a poorly conductive pili, it also stopped the iron oxide reduction. I'll actually talk about that strain more later. We're really interested then to really learn more about the actual conductivity of these pili, and it took some time until Nikhil Melvankar and Dr. Adhikari developed a method for making electrode nanoarrays in which we could measure the conductivity of a single pili filament. And when that was done, it was shown that these pili have what's known as ohmic-like conductivity, shown here, and that this conductivity was more than sufficient to account for the rate that electrons were moving out of the cell onto iron oxides. So this was some initial measurements with a species known as sulfur-reducens. Metalli-reducens, that original isolate, had even more conductive pili, 5,000-fold higher. This is conductivity. It's getting up to close that of a carbon nanotube, and I'll explain later why that is even more conductive. So this is a concept that electrons generated from central metabolism inside the cell move along the conductive pili and onto the iron oxide, reducing iron 3 to iron 2. Now, I just want to point out, though I'm not going to talk about it anymore, that there are other strategies for Geobacter and other microbes to get electrons outside the cell. They can use these electrically conductive pili, and this has been known for some time. This is a paper from John Coates' lab from 2006. Also, some organisms can produce a natural shuttle, which will help get electrons to the cell, or they'll chelate iron, making it soluble, so they no longer have to try to get electrons on an insoluble electron acceptor. In addition to pili, there are cytochromes that can be involved in multiple ways. I just want to acknowledge that. I could spend a whole seminar on how that happens and all the debate about it. I'm not going to go there because I want to focus on the various functions of the protein nanowires. So the nanowires are important for extracellular electron transfer into iron oxide. And I think maybe even more interesting, the nanowires can serve as a conduit for interspecies electron transfer. So we typically refer to this as direct interspecies electron transfer, has a nice acronym, DIET. And we first found this in a very artificial laboratory setup in which Zara Summers put two Geobacter species in a culture bottle under conditions in which they had to share electrons in order to grow. And what she saw was over time, they formed these large aggregates. They're about 1 millimeter in diameter. And it turns out they're electrically conductive. If you stain the two different species, you can see that they're intertwined amongst each other within this aggregate. That's by looking by light microscopy. And then if you look by transmission electron microscopy, you can see between the cells, there's this very dense network of the nanowires that's electrically conductive. So that was a laboratory phenomenon, and we really didn't know if it was interesting, but if it really had any significance. But it's now turning out that it does, because we're finding this diet process is important in environments in which methane is produced. And that's because nanowire containing bacteria like Geobacter and others I'll mention in a minute can use the wires to get electrons to the methane producer, which uses the electrons then to reduce carbon dioxide to methane. And this is interesting because it's a totally different strategy for interspecies electron transfer than had been known for the 50 years leading up to that. So yeah, back in '67, Bryant et al. Made this amazing discovery of how methane systems can work, in which organisms that oxidize organic compounds to carbon dioxide and sometimes other products will use those electrons to reduce protons to hydrogen gas. And then the methanogens will use the hydrogen gas to reduce carbon dioxide to methane. So that's known as interspecies hydrogen transfer. And that's what we all learned was how a methanogenic system works. So we were really interested then, once we discovered diet, to determine whether in any real world situation it might be important, or is it always interspecies hydrogen transfer as we all previously thought. And we're interested in this for a number of reasons. So one is anaerobic digestion, which is the conversion of organic waste to methane. It's actually one of the few effective bioenergy processes that's actually is scalable and works at like a commercial scale. So we were interested in methanogenesis. And I guess I've already mentioned this, that this is just a little animation I made to just give you the idea. You have this one microbe producing the hydrogen, the methanogen uses the hydrogen, reduces carbon dioxide to methane. So we were looking for that. And we worked with Anheuser-Busch and went to a number of their brewery waste digesters and started looking at the microbiology through molecular techniques. So metagenomics, metatranscriptomics, just to try to find out who was there and what genes were they expressing, what might they be doing. And what we found routinely in these anaerobic digesters treating brewery waste, that Geobacter was the predominant bacterium, and the predominant methanogen was known at the time as methanosata. It's now called methanothrix, but it was a methanogen not known to use hydrogen to reduce carbon dioxide. So in further looking at the gene expression in these microorganisms, we could see that Geobacter was clearly using, for example, the ethanol in the brewery waste. Methanosata, surprisingly, was converting carbon dioxide to methane, even though it didn't have the capability of using hydrogen. And we looked at this further in defined co-cultures with methanosata and Geobacter, and could demonstrate that, in fact, it's the diet process rather than interspecies hydrogen transfer. And I think that this may extend a lot further than systems in which Geobacter is important. So a lot of the anaerobic digesters, Geobacter may be less than 10% of the microbial community, but there are, it's well known, other microorganisms such as Syntro. In fact, or Syntrophus, I'm sorry. Syntrophus is basically the poster child for interspecies hydrogen transfer. It was studied for over a decade for that process. And unfortunately, one of the co-authors on this paper, I was very grateful, was Michael McInerney, who his lab did all the Syntrophus studies. And so Syntrophus, the dogma goes by interspecies hydrogen transfer. What David Walker and others found in my lab was that, in fact, Syntrophus was expressing pili, and that these are electrically conductive. And if you place Syntrophus under conditions where only diet is possible, not interspecies hydrogen transfer, it carries out the diet process. So we're now finding that a lot of bacteria that were thought to go by interspecies hydrogen transfer can also do diet. And I think, so there's a question is just how important is diet versus interspecies hydrogen transfer in those systems. The same is true for terrestrial systems that produce methane. We're interested in these because these wetlands are an important source of atmospheric methane, and methane, of course, is an important greenhouse gas. And from our metatranscriptomic analysis, we can see clear evidence in rice paddies, for example, of Geobacter and the Methanosata/Methanothrix carrying out this diet process. So I think there's still a lot of work to be done, but diet is now a new paradigm for thinking about how these methanogenic environments work. And we're trying to think of like, why might diet take place rather than interspecies hydrogen transfer? And I think one reason is just speed and stability of electron transfer. So if you have two centromes or microbes feeding methanogens, diet works fast. Once the electrons get to the outer surface, it's fast. Look, whereas hydrogen is diffusive, right? So it goes slow. So and also this supply of hydrogen can be broken up many ways that may interrupt that flow of hydrogen, whereas once the cells have established that electrical connection, that's really a tight interaction between the two. So there's an advantage to diet, but there's also a cost. So in my analogy for this is electrification of human civilization. So we used to deliver energy in a very slow, diffusive way. This is coal delivery. Energy delivery, of course, was greatly improved by electrification, but there was a massive investment in producing the wires and the other infrastructure for electrification. And I think that's the trade-off between diet and interspecies hydrogen transfer. So the question is, when is one important versus the other? And that's still not fully worked out. But understanding this investment that the microbes have to make in the wires, we have found ways to basically alleviate that cost. And that is by adding conductive materials. So these would be inexpensive materials like activated carbon, carbon cloth, biochar. They're all electrically conductive. And if you put them into one of these systems, you'll make it trend towards diet. And the reason for that is the microbes no longer have to make the nanowires. So you can see in the scanning electron micrograph, instead of now being connected. So this is a defined system with Geobacter and methanogen known as methanosarcina. You can see that they're not even like close to each other. What all the cells are doing instead now is they're plugging into this conductive surface. So what's happening is the bacteria that are oxidizing organic compounds are pumping electrons into the conductive surface, and the methanogens are pulling electrons out of that conductive surface, reducing the carbon dioxide to methane. And this turns out to just almost routinely in all anaerobic digesters speed up the production of methane, typically gives higher yields of methane, and it can stabilize the methane producing community to introduction of toxins or overloading the system with too much organic waste. So there seems to be a practical benefit to switching the system towards diet. Okay, so just like the microbes can donate electrons to the conductive material like an anaerobic digester, it turns out that they can also, and this was actually discovered first, can exchange electrons with electrodes. And so they're doing the same thing that they do with other cells or with iron oxide, oxidizing organic compounds of carbon dioxide, but now transferring electrons to an electrode surface. And if you connect that to a cathode, you can get electrical current. And many, many microorganisms can produce electricity. So far, though, it turns out Geobacter produces the highest current densities. And in order to get high current densities with Geobacter, you have to get it to produce the nanowires and form a really thick biofilm, which turns out to be electrically conductive, and it's only electrically conductive if Geobacter is expressing conductive nanowires. So that's really the only way to improve current production is to get Geobacter to make more wires. So there are some practical benefits of this. This was an invention first discovered at the Naval Research Lab by Lenny Tender, Claire Reimers, and others. where you can just bury a graphite electrode into an anaerobic marine sediment, connect it to an aerobic electrode in the overlying aerobic water, and you'll get a flow of electricity. And what we found was this is because geobacters will colonize this electrode. So they're doing what they always do. They're oxidizing organic compounds of carbon dioxide. But now instead of transferring electrons to another cell or to iron oxide, they transfer electrodes to the electrode. And you can get a small amount of current this way, which is useful to the Navy for locations where it would be difficult to go back and swap out batteries. You can power some types of electronics. I think one of the great benefits of the so-called microbial fuel cell has been it's turned out to be a great science fair project for teaching microbiology. And people used to make their own microbial fuel cells. Now, if you're kind of late and your project's due in a couple of days, you can get on Amazon and order this, it's called a mud watt, and throw some mud in it and wait a couple of days and it'll read out electricity, or you can on your phone. And so it's a good science fair project. I actually helped my daughter set one up this morning. You can get a Geobacker plush toy if you want to go with it. And I think it's great for teaching microbiology for K through 12. But the big, the push is for like other applications. And we worked on this for quite a while. We had a project funded by Toyota. I like working with Toyota. They make 100-year plans. I thought I'd be set for a good part of my career for funding if I could work this out with Toyota. And Toyota came to us with this strategy of, well, could we make at least some of the power for vehicles? So this is where we wanted to get, but this is where we're at. So the issue, it's not the microbe's fault. I mean, we can engineer Geobacter to make many more wires and higher current densities, but the electrodes have to be really small in order to realize that benefit. And it turns out there's electrochemical limitations, and Jason could explain this much better than I can. They're preventing the scale up of these systems to produce a lot of power. And this is, I think, really the challenge that many people are working on and hopefully will solve. But I have nothing to contribute there until somebody engineers a better system. That's the best we can do for current production. But there are a number of applications that don't require a lot of current production. I really like this idea of using electrodes for bioremediation. And I should have got a slide from Abraham Esteves-Nunez, for example, has, I don't know, 1 or 2 companies where they're successfully making wetlands that are filled with electrodes and treating wastewater very successfully with this concept where the contaminants are oxidized and using the electrode as the electron acceptor. We can monitor rates of microbial activity using these sensors or using this technology, because the amount of electricity you get is related to how fast microbes are metabolizing organic matter. So that works, and we've used that in some environments. And you can also then get microbes to go, I think it's interesting to know, not just to make current, but you can feed electricity to microbes. So if you poise this electrode at a low potential, electrons will go the other way. And this was first seen by Kelvin Gregory and Daniel Bond with studies with Geobacter, so we could drive its respiration. So again, kind of just a laboratory interest, and some practical applications have been developed. I think the question is whether they can be scalable. So for example, Kelly Nevin and others developed this process known as microbial electrosynthesis, where you can use renewable electricity, say from solar panels, to again go back. If I go back, you're using that electricity to feed the microbes. Now they're reducing carbon dioxide to produce organic compounds. We had a project making butanol, for example, and it all works. But again, it comes back to this scalability issue. And again, I'm really hoping Jason and other engineers will be able to figure out how to get these systems to scale, because right now they are not commercially viable. And if it would scale, for example, microbial electrosynthesis has a number of potential advantages over the way, biomass-based fuel strategies that we have right now. So we'll keeping our fingers crossed on that. And there's also some bioremediation capabilities. You can use electrodes to reduce uranium. I mentioned treating those environments before, and also chlorinated solvents. So some of these are really just shown as proof of concept so far, and some are actually being employed. But what I'd like to talk about for the rest of the time, how are we doing on time, is no longer dealing with living systems, but dealing with these electrically conductive protein nanowires. And the reason for this is they are a new type of green or sustainable electronic material. And we've been saying this for a while. Just fortunately, earlier this year, we were able to finally get out some of the papers on this. And I mentioned this in the beginning, and I'll describe some of these applications. I'm primarily going to focus on the sensing application. But the idea again, and I'll show you what we did to make this E. coli strain that can mass produce these wires much faster and much better than Geobacter. We harvest the wires and then use the wires as an electronic component. So I first got interested in this idea of like sustainable electronics just because I was reading a lot about electronic waste. We know that waste is piling up. There's other costs that I learned from the Semiconducting Research Corporation, for example, the high energy cost that goes into mineral extraction, like for silicon devices. And it has, I thought it was just, oh, you can make this stuff from beach sand. It has to come from a very pure silicon deposits, and eventually we'll run out of those as well. And making silicon wires really involves nasty processes, and so do making things like carbon nanotubes or polymer wires as well. So I kind of came at it again from this environmental aspect. And though I never expect this environmental aspect, which we published earlier this year, which that the protein wires are giving us a totally new sustainable way of producing electricity. And basically, I'm not going to go into all the details. Frankly, some of the mechanisms we don't fully understand yet, but you can make a thin film of these wires, a 10 micrometer or less film. If you organize the electrodes in the proper way and just leave this out on the benchtop, it will produce electricity. And what we do know is that there's a decreasing water content gradient and increasing protein concentration gradient. And this gradient results in a current flow through the biofilm. And if you connect these electrodes, you'll get a current flow through a device. And it's thought by many that water absorption and then evaporation yields energy. And that appears to be what's driving this device. So there's still a lot more to be done on this. The advantages of this over solar, for example, it works 24/7 in the dark. Same thing with wind, it's not intermittent. The right now, as with most new technologies, the power output is low, but you can use this to generate power to light up electronics. And this work is being done in the laboratory of Jun Yao, my collaborator at UMass. Xiaomin Liu is a graduate student that first discovered this basically serendipitously while he was making sensors. And they're working up on the scaling. But I'll show you they've already come up with some practical applications as well. But yeah, I'm primarily going to focus on the sensors because I think this is where the protein nanowires may have a big advantage and possibly a large market. And some of the advantages over their competing materials like silicon, conducting polymers, or carbon nanotubes. One is they have function already, I'll show you, but they can be more readily functionalized for broader sensor applications than silicon. They have a dynamic response. They're better than silicon for biomedical devices because they don't dissolve in water or bodily fluids like silicone does. They're biocompatible. I'll show you how they're mass produced. And I've already mentioned the energy requirements, no toxic chemicals for making them, no toxic components in the final product. They're biodegradable. We get really uniform production. And despite the fact that they're a protein, they're super robust. They are stable under conditions typically used to make electronic devices. And I'll show you some of the other advantages as well, for example, self-powering. But I think in order to explain kind of where we see the protein wires going, I need to give a bit of idea of what we think we know about the protein nanowires. Actually, there's several types of protein nanowires that can be produced from Geobacter. Some are comprised of cytochromes that their biological function and also their electronic function have not sorted out yet. So I'm going to talk about the wires that I've basically been talking about all along, which are homologous to the type 4 pili that are found in many microorganisms. And what's great about type 4 pili is that they assemble from a single monomer protein. So in Geobacter, that protein or peptide is about 60 amino acids. So it's a small protein, only one gene to then influence the properties of that wire. And it's thought, and not fully confirmed by structural analysis, but it's thought that as these pili filaments components come together, the aromatic amino acids within different components pack together tightly enough that this yields aromatic to aromatic electron transport, potentially through a core in the center of that filament. And we can see the importance of the aromatic amino acids simply. I'd mentioned this strain before that made the poorly conductive pili. If you think aromatics are important, what we did was we swapped in alanines for aromatic amino acids. We got a strain of Geobacter that still made the filaments and it still expressed its outer surface cytochromes. For example, these black dots is immunogold labeling for OMCS, an important C-type cytochrome. So it's still making wires, it's still making cytochromes, but the wires that we harvest are poorly conductive, as shown here. So that tells you aromatics are important. Another way, we know that aromatics are important, as I mentioned before, metalloreducents has wires that are 5,000 times more conductive than sulfur-reducents. And you can see when you look at the amino acid content of metalli versus sulfur, that metalli has way more aromatic amino acids, which seems to account for its higher conductivity. And once you realize that these aromatics are important in conductivity, you can start then looking through genomes of isolated microbes or even through metagenomes to start finding other microorganisms that might have conductive pili. And David Walker did an excellent job on this, developing this theory and then demonstrating it, looking at different bacteria, including this syntrophis, which I mentioned earlier, which is, I think, really key to understanding diet and methanogenic systems. And these type of studies go on, it just keeps expanding the diversity of protein wire microbes. Here's another example of that. If you think aromatics are important, you can start looking at other microbes. This is Archaea now, Methanosporidium hunkeatii, for which the Nicole Pullett had already solved the structure and published that. Our colleague Eric Martz looked at this structure and saw this, again, a core of aromatic amino acids in the center of the filament. And sure enough, when David Walker measured the conductivity, here's the filament coming off the cell. When he measures the conductivity, this is the filament. This is a 10 nanometer diameter filament, so it can make fatter wires. It's conductive, and in fact, it has a higher conductance than the Geobacter. So there's a lot of different wires out there, and knowing that aromatics are important, we can start to now make synthetic pillin genes. So for synthetic genes to make synthetic pillin monomers which assemble into the wire and add, for example, in this case, we swapped in tryptophan for two of the existing aromatic amino acids. Tryptophan has extra rings. And surprisingly, in this case, the wire is smaller for reasons that aren't quite worked out. And I think we would have been happy if we had doubled the conductivity of the wire, but we actually increased it by, I think it was like 500 volts. So this is up near conductivity of a carbon nanotube. So you can tune the conductivity of these wires over at least 1 million, maybe 10 million fold, could probably go greater if we needed to. The reality of it is for sensors, you sometimes want to be down more in this semi-conducting range. So the native wires function as sensors. And this was first seen by Mikhail Malvankar and others, that there's a strong response to pH. So this is for an individual wire that, and you can see as the pH decreases, this is a log scale, greatly increased response. And so this led us to this sensor concept. We also now know based on the electricity generation study, that we can make different sensors that will power themselves. So this is an example of, it's just the lab prototype, of course, but it's a self-powering wearable sensor. These sensors can also be used for measuring humidity, and this would be potential importance for, say, monitoring respiration or hydration of skin. Again, these are all proof of concept studies. I do like this humidity sensor, though, because it also, as we showed in this paper, can be used as a trackless sensor. And certainly nowadays, when none of us want to be touching anything, this might be useful technology. And the native wires also have some response to ammonia. So, and actually quite selective versus other volatile compounds. And yeah, this is just the comparison. So the response time and detection limit are actually quite good for the native wires. But I think what we're really interested in is tuning wires to be specifically conductive or responsive to different analytes. And what the key here is that we can functionalize the protein nanowires in a way that cannot be functionalized, or difficult to functionalize a silicone wire or a carbon nanotube. And that's because we can, through genetic manipulation, produce wires that display protein ligands, as shown here, these blue sets of blue balls. They're specifically displayed on the outer surface of the cell. And the reason you would want to do that is it can improve sensing. So here's a strategy. You just take a normal pillin gene, add a little bit of extra encoding on for the carboxyl end of the pillin monomer to add that peptide ligand that you would like. And it's pretty well known, it's well established in the literature, that you can design short peptides of, say, 5 to 10 amino acids that will specifically bind a wide diversity of analytes. So we make that synthetic gene, we put it in, we first did it in Geobacter, we can now do it in E. coli. So it expresses the wire with those ligands on the outer surface, and that's basically the new sensing ligand for the sensor. So just to show you how we know we did this, was we first put on peptides that are easy to analyze. For example, a histag is 6 histidines, and there's antibodies available for that. So we made a gene had a histag, and this is a strain that is producing some of the wild type and some of the histag pillin, and you can see that all the wires that are emanating from the cell are immunogold labeled for the histag. That's these black dots here. They're just very different variations of that. Oh, I'm sorry, immunogold, also using a nickel gold reagent that's specific for the histag. You can put multiple ligands on a single wire. This is using something called an HA tag, for which there's also an antibody. So we were able to look at here's the histag now with we attach a big gold particle to that antibody, small gold particle to the HA tag, and you can see the multiple tags that are on the wire. We can control the stoichiometry of one tag or multiple tags using genetic circuits that control how much of that specific pillin of interest is produced. And fortunately, adding on that peptide ligands has no negative impact on the conductivity. In fact, for the 2 that we made, or at least one of them, it actually slightly increased conductivity. So that was lucky. So again, so the idea is if you have these ligands sticking off of the wire, they're specific for analytes of interest. When this analyte binds, because of the near field interactions, this will change the conductivity of the wire and therefore change the electrical output of the sensing device. So to make a lot of these devices, it really doesn't work to just put flat wires out there without any covering on them, so to speak. So we've been working to make electronic composite materials in which you put these wires into a polymer, and this is the initial proof of concept studies that shows that this works. This is using the polymer polyvinyl alcohol. You can put the wires in. The more wires you add, the more conductivity, up to a certain point. This is what's known as a percolation threshold. Adding more wires doesn't really improve the conductivity. But anyway, we can take a polymer that's non-conductive, now make it a conductive composite material. And it still not only has its conductivity, but it has its sensing functions, for example, the pH sensing function. And can make these then within a range that's a reasonable range of what's known for other flexible conducting composite materials. Oh, so yeah, this is one other thing that we can do with the wires to try to modify them for electronic devices. So normally those thin films of the wires are all randomly distributed. But if you treat these wires with various organic solvents, they're still stable, they retain their conductivity. And if you do it right, they'll all line up. And you can see that here. So you can make tapes and ropes and different structures that my collaborators in material sciences like Todd Emmerich are very interested in, and they're just seeing what can be made with these wire materials. So for my collaborators and for any potential future commercial success, we can't make wires with Geobacter. Geobacter is an anaerobe. For most people, it's difficult to grow, so probably no one's going to work on these other than our lab if we didn't make this simpler. And frankly, we needed more wire. So the concept was to take a strain of E. coli, a typical laboratory strain that, say, high school students even work with, easy to grow, put in the genes to allow it to make pili, and then set it up so it would be easy to switch in pillin genes so you could make different wires. And this fortunately worked. So it enables rapid mass production of the wires because E. coli can be grown aerobically. We just grow it on the benchtop or in big fermenters. And what I really like about this is that there's genetic tools for Geobacter, but nothing like the extensive synthetic biology toolbox that's available for E. coli. So this is going to give us it already has the ability to make even more sophisticated types of wires using various genetic circuits and mechanisms for gene control. So this is basically what it looks like. There's this insertion of genes to make pili. This is the monomer gene this is the one we want to take in and take out to change what type of wire we make. And we can just physically shear off these wires, collect them simply by filtration, and the wires look just like Geobacter wires. So I'm freely giving this strain away. I'm hoping that maybe we can make protein nanowires into a little bit of a field here. So now here's the production process. Anyone should be able to do it. If anyone's interested, please contact me. So I hope it's clear that I know I focused on Geobacter, but there's a lot of microbes that have nanowires. They have long-range electron transport to other cells, minerals, biofilms, but they also show promises of electronics, and we've already for sensors, flexible composites, and I really like this concept of sustainable energy harvesting from the ambient environment. Anyone should be with wires now, and which hopefully other people will do, because there's really a big need just on the basic research side of the mechanism of electron transfer, and more research needed in applications, and also biological distribution and function. So again, thank you very much for your attention. Oh yeah, there's the Geobacter toy lighting up. Thanks so much, I really appreciate it. Hello?
There you go. Well, thank you, Professor Lovley. That's a really fascinating talk, covering a lot of grounds and from different aspects, fundamental science to applications. So there are a few questions. I'm gonna just rephrase that and see and sort of try to group them. The first question is from Winnie Coela. So great talk, fascinating, and using some of these microbial electrochemical methods, there are opportunities to bioremediate toxins in the human body. Say, for example, disease prevention and other things, or remediate toxins in the brain, maybe precursors or like people doing drugs, I assume, or brain tumors. great question. That's interesting.
Yeah, it's a great question, which I've never thought of. So, I mean, I think there's growing interest in I guess what we call the electromicrobiology of the gut, for example. And there's possibility, others have found microbes that are electroactive in the gut. It's a very unexplored area. And I think, the questioner, I think you've maybe struck on a very good new research topic, because the possibilities there are totally unexplored.
Yeah, yeah. There's an early question from Deepak Pant, who is certainly a very active researcher in our field, asking, you mentioned about sort of the biology as one side of the, I guess, important factor for actually system scale up on applications, and any other factors that may actually limiting, I guess, the scale ups. I'm not sure that's a question for you or for me, but if you can give a few thoughts, that'd be great. I'll be happy to add a little bit. Yeah.
I really rely on Deepak and you and others in the field. I'm hoping you all are going to solve it. Yeah, maybe you should say something about that.
Yeah, I mean, there are a lot of people working on this, and certainly we all actually build on the fundamental understanding from Professor Lovley's work and others' work. I feel a fundamental challenge we are facing is related to the uniqueness of electrochemistry. That's sort of something we are working on, because electrochemistry, whether it's microbial or abiotic, is a sort of 2D reaction. I mean, it's basically related on the surface, and we always limited on the surface area. How do we scale up, right? If you have to make a big one, you have to buy very compact electrode. So if we can actually sort of expand that 2D reaction into a 3D, whether using flow cell or other configuration from engineering perspective, I think that can bring a sort of a new application potentials into this field. So Jose Avalos, who is a faculty in our center and the CB department, asks you, have you used protein wires expressed on E. coli to control any function of E. coli cell, for example, growth and other aspects?
That's a great question. So no, the short answer is no. I can explain why, I think. So even though we have E. coli expressing the wires, there are other electrical connects to get electrons either to the wires or from wires into the cell. And I think that's one of the next step. These are, it's well known in Geobacter and other organisms like Shigella that they have cytochrome-based conduits for electron transport across the outer membrane. And somehow the wire and the cytochrome conduit need to get linked together. And I hope to do that in E. coli, just to see if our models for how microbes actually are electroactive are correct. But so far, no, we haven't been able to do that.
Great. Well, there are two windows I have to deal with. Sujit Datta, who's also a faculty, asking that are there ways to produce protein nanowire in a larger scale, and does it require growing a larger culture, and how do you remove those nanowires from sort of mass process?
Yeah, those are all great questions. So it's well known that E. coli can be grown at really large scale to make commodities. I mean, that is already done. So you just need a larger fermenter to do that. Then the rest of the scaling is you need to remove the wires, which we just do by stirring. I mean, the wires are not that well stuck on the cell, and then we run it through a filter. So I think all of that is scalable. We have had no need to run anything larger than like a 5 or 10 liter fermenter. But based on what other people have done with E. coli, it should be scalable. I think the other question that people ask is, why do you need the microbe at all? Couldn't you just take that pill and protein and work out conditions to make it happen without biology at all? And unfortunately, the answer is no. You get a mess. And other people have tried designing wires to be made in vitro. They just don't have the scalability or actually the quality of the microbes. So right now we're still dependent on the microbes, which just seem to be they have really exquisite machinery for making these wires in a very specific way.
Yeah, wow, a lot of questions pop up now. So from Robert Austin asks, I know you've done this, Derek. So have you measured the conductance versus temperature? So that's a fundamental sort of, I guess, parameter on the conductivity. If so, what do you see the results going to be, I guess?
Yep. So yeah, that was done early on by Nikhil Malvankar. But I want to make it clear, so it hasn't been done on an individual filament, but it has been done on like thin films. So, or at least his work was on thin film. So what he saw was as you decrease the temperature, then the conductivity would, at a certain point, start to increase. So that was actually led to the whole idea of aromatic amino acids being important, because Nikhil interpreted that as metallic light conductivity. There's another lab that looked at the temperature response and said with individual wires, they got a different response. I just didn't really want to get into this. So there's this big debate about the mechanism of electron transport, whether it's metallic light or electron hopping. I'm remaining agnostic, because both for the biology and the practical applications, as long as they're conductive, it's okay. But I think I'm sorry, I'm starting to ramble. I think it's a really important question to work out the mechanism of electron transport. Right now, it's, I would say, a matter of debate.
Yeah. So another question is from Aaron Langer. So he asks, what about the sort of the magnetic fields, right? So you can induce current in wires with a field, right, without direct contact. What can we learn about this sort of nanowire properties by studying the induction response? Or do you see any actually other application related to, say, induction devices on that aspect?
That's possible. I mean, it's these are things I really don't think about. I'm just a microbial ecologist, so this is why I think with the E. coli strain, I'm really happy that I'm hoping more people that are more into electronics will get some of these wires, or we'll make them for you if you have an interesting idea, and try out some of these ideas. Because the short answer is I have no idea. I wish somebody would try it. Great.
That's why, again, I think there's great collaboration opportunities over there. So let's go back to biology questions.
Okay.
Are there cases where organisms that do not form nanowires coexist in a biofilm with Geobacter and use Geobacter's nanowire sort of symbiotically? That's from Peter Jeffy.
Possibly. And I think that idea has been proposed. And so certainly in the diet process, the methanogens, well, some of them have wires. So it's confusing. But some microbes that don't have wires can electrically connect with a Geobacter or other microbe that has a wire. So I think there's many different types of interaction. And then once you get into a biofilm, it gets hard to sort it out. But I think what the questioner is proposing is certainly possible.
Yeah, wow, we even have questions from Denmark. So Paula is asking, would another aerobic electroactive bacteria, probably anaerobic, like Mastell-Coccus, express Geobacter nanowire instead of E. coli? Well, aerobic, I think that's what she means too. So basically, is there other, like a better host to actually getting more wires, I guess?
Yeah, so I guess it depends what you want. I mean, I think for just mass production of wires, probably E. coli is the best, unless you could get yeast to make them. But I think for commodities, most people use E. coli. And, but I mean, maybe what part of the question is for like, if you wanted to make an electroactive microbe that you could grow aerobically, there will probably be better choices. Well, you could put it in Shionella, for example, you know, which can be grown aerobically. And in fact, David Walker did that. I shouldn't probably said that he hasn't published it yet, but you can put it in a lot of microbes. It's just what your goal is. If you're trying to make electrical connections or if you're just trying to make wires.
Mm-hmm. Another question is about the cytochromes. So as you're aware, like Nicole and other folks and sort of recently published findings that so at OMCS sort of also formed this sort of what we call nanowires, or not necessarily pili. There's certainly different opinions and people still trying to figure that out. But do you see like a sort of other type of nanowires form that can be also harvested in sort of a same level of, I guess, type of applications of this protein nanowires? Yeah,
that's a great question. So, I mean, we also reported on the OMCS nanowires as well, but we don't like, let's see, do I still have screen sharing on or?
Yes, you are.
Yeah, I just thought I could, if you go back, maybe this isn't like zoom back enough, but when you label the pillins with a peptide tag, for example, in Geobacter, at least under the conditions that we grow them, all the wires are labeled with pillin. So there's no evidence that that under these native conditions that OMCS wires are being expressed. So it's not to say that they couldn't be, and you certainly find them in preparations of wires. So I think the questions, well, I have multiple questions on the cytochrome wire if we have time. One is, I don't know how to make them because, and I can't, there's no way to make them in E. coli because, and even for Geobacter, we don't know how, if they're extending from the cell, how are they made? And we don't know their biological function yet. So I think it's a great question. We're focusing on the pili, and I think part of it is also just, it's so easy to express them in E. coli. It's so easy to functionalize them for all the practical applications. It seems to me that the pillin-based wires are more useful for applications than the cytochrome wires. But I think the biology of, all biology is interesting. Cytochromes we've been studying forever. I definitely would like to get all the role of all these proteins all sorted out, but it's not yet. Yeah.
Well, there are a few questions left. I'll just try to group them together in just in observation of time. So for the Zara Summers, you probably know her. Asking any advances in understanding the role of diet in ANME, anaerobic methane oxidation, I think, right? Archaea consortium, right?
Right. The question is, what is the understanding of how
of diet in sort of a methane oxidation or anaerobic methane, I guess, metabolism consortia?
Right. So there's again, like everything, there's multiple theories on that. So some groups propose that wires are important. The other electrical connect that's clearly on the outside, and some of these ANME consortia are multi-heme C-type cytochromes. Mm-hmm. So it's, again, the problem with ANME is you can't grow the microbes independently in the lab. You can't do genetic manipulation. So everything is observation. So it's kind of, right now it's at the just so story kind of stage because we can't do the functional studies with the ANME yet. So, but those are the two competing ideas. And I've written about this before. It's just cytochrome electrical connect, like Victoria Orphan proposes, or Wegner and others in Germany propose a nanowire electrical connect, or maybe it's both, or you know, nobody really knows for sure because it's all observational.
Yeah, yeah. I mean, so this is really a new area, right? There's people have so many questions on the how do you deposit wires, how do you increase the production? I think that's more engineering questions. I won't post them to you. So we have a question from India. There possible to completely reduce iron 3 to iron 0, I guess, for a single sort of cell or culture, I guess. I don't know, that's probably on the remediation side. I can answer it for you, that's yes. And from the, just so curious, on this sort of protein harvesting, wire harvesting side of it, I mean, do you see any engineering approach, like people, I mean, in this, say, like a deposition or like other ways to actually, again, when you grow the wire, like how do we actually put them onto sort of certain surfaces to make I guess, conductive devices, right? So sort of things like that.
Right. So that's a great question. And again, I'll just give you my superficial understanding of it. For a lot of the initial devices, we just what's called drop cast them, which just means drop the wires onto electrodes. But one of the advantages of being able to modify the wires is we, in theory, can make wires that have a sticky end on one end, so it has a slightly different composition on one end than the other. And my collaborator, Jun Yao, who's made all these devices in his lab, he's a silicone nanowire expert. Alignment was what he worked on in Charlie Lieber's lab. He knows all about all these things, and he's got some really good ideas for getting these to line up when we need to. And sometimes we just need random, it doesn't matter, a random array of wires is fine. But that's something that has not been done yet, other than what I showed, the tapes by organic solvent. There's a lot to be done. And again, what I'm hoping is other people that are interested in the electronics and devices will get in this field, because I'm basically done. I'm going back to mud. I kind of did my job here. I can make people can make wires, or we'll make them for you with E. coli. It's really simple. Undergrads can make them. We have undergrads make them all the time. So I'm hoping people will, if they find this material useful, will try it out and try some of these things.
One last question, Derek. How about Shrinella or other sort of I mean, that's not necessarily that's a different mechanism to a certain extent. Or other like bugs you think probably can also be harvested for their wires? Yeah.
So there's so many now out there that have these pillin-based wires. So Shrinella has these what are they called? Membrane blebs that are, it's a mixture of cytochromes and extracellular or outer membrane lipids and whatever. I can't see how to work with those or to mass produce them, because you get maybe 1 or 2 blebs per cell, and how you make a uniform material. But so Shrinella, I'm not that hopeful for, but they're nanowires or blebs. But there's so many wires in the microbial world now. We just started with the Geobacker one. I think almost all the microbes like Geobacter are hard to grow. So if you want to study the biology, yes, you have to study that hard to grow microbe, but you can express any of these wires in E. coli now. And so nobody's fixed on any one specific wire. Pick your favorite pillin protein, or it can be an Archaean gene, and express it in E. coli and make whatever you want. I think that's what I'm really excited about.
Yeah, I agree. Well, great. So I want to thank you again, Professor Lovley, for the great talk. Everybody's enjoying it. And hopefully there will be follow-up conversations to really move this exciting new field forward. Thank you so much. Appreciate it.
Thank you, Jason. I really appreciate it. And if anyone has questions, feel free to email me, or happy to talk to you online later. And again, thanks so much. Really appreciate the opportunity to get this story out. Thank you.
Thank you, Derek. And thanks everybody for attending. We made a record actually on attendance today. Great job.
Thanks. Okay, bye.
Bye.